And thanks for joining us for another fantastic Bioprocess Online Live. And I, too, would like to thank Precision Nanosystems for supporting this digital event and making it available to the Bioprocess Online and Life Science Connect communities. I'm Matt Pillar, Chief Editor at Bioprocess Online, host of the Business of Biotech podcast. And today we're talking mRNA with two guest panelists who bring more than 60 years of combined experience in ATMP, CMC, and technical leadership. Jeff and Thomas are looking at each other right now saying, there's no way that combined we can be worth 60 years of experience, but you are, believe it or not. Dr. Atkinson is here joining us from Omega Therapeutics, where he serves as Executive Director of Process Development and Analytical Sciences and Technical Operations. Dr. Atkinson began his career with the Southern Research Institute after earning his Ph.D. at the University of Alabama at Birmingham. He's since directed technical operations at Brookwood Pharmaceuticals, Evonik Industries, Ipsen US, and now Omega, in addition to doing some private consultancy. Also joining us today is Combined Therapeutics Chief Scientific Officer, that is Dr. Thomas VanCott, who earned his Ph.D. at the University of Virginia before serving as captain in the Medical Service Corps at Walter Reed Army Institute of Research, then directing operations at the Henry M. Jackson Foundation for the Advancement of Military Medicine. Thank you for your service, Dr. VanCott. He then joined industry with ABL Industries, where he advanced to president and CEO before joining Combined. Drs. Atkinson and VanCott, welcome. Thank you for joining us. We're honored that you're here to share your time and expertise.
Well, Matt, thank you. It's great to be here. I'm excited about this session and the great audience that we have today. So I'm looking forward to the discussion.
Yeah, same. Thanks, Matt, for introducing us. Really excited about the discussion today, and great topic and great audience that I understand we've got a lot of people on the line, so it should be wonderful.
Yeah. So this point is directed to that great audience. Thank you for joining us as well. And I'd like to reiterate that this next hour is yours. Drs. Atkinson and VanCott have agreed to take some Q&A, so please do use that Q&A box early and often. So let's jump into it. I want to start at sort of a high level, Dr. VanCott, with you, and I'm looking for some reflection on where we are today and how far we've come recently in terms of delivery beyond the liver.
Well, Matt, that's an excellent way to start. I mean, I think we could dedicate a full hour, if not 1 hour. We could dedicate multiple sessions to this topic. The last few conferences.
All right, you've just gone on record. I'm taking that as your commitment to do a multi-part series with me.
There you go. I mean, the last couple of conferences I went to, this was one of the main topics, And, I kind of came at it a little bit from the gene therapy, organization where, we were using viral vectors to deliver our cargo. And, and there's a real push to try to get away from viral vector delivery and get the non-viral. And that's kind of where we are. That's what the COVID vaccine is. It's a non-viral delivery. In this case, it's a lipid nanoparticle. But, these go predominantly to the liver. So the question is, if we want to start delivering therapies to sites outside of the liver, how do we do that? Well, I mean, the way I look at it, there's really two predominant ways to do that. One is the delivery vehicle itself, whatever that might be. Can we develop a delivery system that will go somewhere besides the liver? But second, you could engineer the mRNA itself. You could engineer it with some on or off switches that would allow protein expression in some cells and block it in other cells. So those are sort of the two major categories. But if you look at the delivery vehicle itself, I mean, we're all familiar with lipid nanoparticles, right? That's what the COVID vaccine is being used. That's what predominantly a lot of the research is done, but there are other nanoparticles that are being developed as well. And we won't go into detail, but there's protein nanoparticles, there's DNA nanoparticles, there's exosomes, and they all have some unique properties. And a lot of them have properties that they don't go directly to the liver. So those are being developed by a lot of different biotechs and scientists to try to take advantage. For example, you can make these protein nanoparticles that use endogenous proteins that on their own will selectively go to particular cells and tissues. So they'll base their non-viral delivery on that particular type of protein.
Mm-hmm.
But also, even if you look at the lipid nanoparticles, there's a lot of research being done to get those to go to areas other than the liver. So there's passive ways, and when we say passive, that's really modulating the lipid composition, adding an additional lipid or so that can get it to be directed outside of the liver. So there's a lot of work being done, a lot of preclinical studies. They'll do like DNA barcoding and track where their lipid nanoparticle goes, and then they can modify. Some use AI as well to try to modulate the lipid compositions to try to get that particle to go exactly the tissues and cells they'd like to go. But another way is actually adding moieties to the surface of the lipid nanoparticle. And these moieties could be small molecules, they can be peptides, they can be monoclonal antibodies. And those particular moieties will bind specifically to cells in other tissues, right? So if you want to go to the lung, you may, attach a moiety on it that would be specific for lung cells, et cetera. So that's a big area of development as well. Of course, there's complications. It complicates the manufacturing because now you have to produce that additional moiety. And then you also have to make sure that you don't impact, the way that particular particle is taken up by the cell or endosomal release. But I mean, suffice to say, there is many papers published all the time, many companies that are really looking at getting much more precise targeted delivery of their particular transgene and trying to eliminate any off-target targeting. So
Yeah.
really exciting topic.